All right. Hopefully everyone is back and you enjoyed your break. So, as people and experts in fetal therapy around the world are starting to think about the next phase of fetal therapy, without a doubt, one of the areas that individuals and centers are particularly excited about is stem cell and gene editing therapies, and moving those therapies that are quickly becoming the mainstay or standard in pediatrics, moving that to the in utero or fetal world. And we're really, really excited about this next group of speakers under this next section, and starting with a world-renowned researcher and leader in the field. So Dr. Maria Grazia Roncarolo is here with us tonight, and thank you, Maria. And she's really a pioneer in cell and gene therapy for a variety of diseases. She's a world-renowned expert in immune regulation, and is the director of the Institute for Stem Cell Biology and Regenerative Medicine. She leads efforts both on translational medicine side and clinical programs, and really has been thinking about this and leading this for a very, very long time. We're really excited to have you, Maria, and we look forward to your presentation. Thank you.
Thank you. And can you see my slides?
Yes.
Wonderful. Well, first of all, thank you for the invitation. And it's really a pleasure and an honor to be here today. And I have to say that when I was putting my talk together yesterday, I realized that what I am still doing today in the space of immunological tolerance, stem cell transplantation, and gene therapy really stem from a failure early on in my career, in which I made the wrong assumption that I could cure genetic diseases with fetal therapy. So I want to take you through this journey because it's quite interesting how sometimes a failure really brings you to a new field. So I, of course, work as a pediatric immunologist in genetic diseases of the immune system and the blood, and try to fix these diseases with hematopoietic stem cell therapy. And indeed, the very first disease that I work on was a severe combined immunodeficiency, what at that time we called these children bubble boys, because these children have a profound impairment in the immune system, in the adaptive immune response, which lead to fatal infections if we don't protect the children with the bubble to really create a barrier with the external world. And at that time, when I was studying these children, I was a young pediatric resident, we didn't have any chance to cure them unless we had an HLA identical donor. So I went to France because in France they had a very innovative program where they were using fetal stem cells for SCID patients that didn't have an HLA identical sibling. And indeed, these were the majority of the patients. So we cure several patients with severe combined immunodeficiencies using fetal donor stem cells. But what it became clear is that we still had to deal with graft versus host disease as soon as we were using cells from, older fetuses, or we were dealing with rejection when the host was not really a profound immunodeficient patient. So at that time, in a very naive way, I went to my mentor, Jean-Louis Touraine, and said, look, we put these children in a bubble. Why don't we use the natural bubble? Why don't we treat them in utero? And Jean-Louis Touraine was really a pioneer, because he was working, he worked with Bob Good at the Memorial Sloan Kettering. So he said, basically, of course. And there we did. And the first patient that we treated, a bare lymphocyte syndrome, a deficiency in class 2, MAC class 2, was a big success. But then the second child was not such a great success, was also a SCID. And then when we moved from immunodeficient fetuses to competent fetuses, like fetuses with thalassemia, or with metabolic diseases such as metachromatic leukodystrophy or adrenal leukodystrophy, we failed. We didn't see engraftment. In some patients, we had microchimerism, but we never reached a therapeutic benefit. So that was quite devastating. And basically, we proved at that time the in utero hematopoietic stem cell transplantation was feasible, was safe for the fetus, although we occasionally had some fetal death. But overall, we really showed that unless we use an immunodeficient fetus, we didn't achieve benefit because the transplant didn't engraft, was rejected by the fetus. So, to make a long story short, I spoke to Paul Berg, went to work at the Next Research Institute in California Avenue, and then I met Mike Harrison. And Mike said, no, it works. It's just because you use fetal stem cells and the number of the stem cells is insufficient. But if you go with a higher number, you will overcome the allogenic barrier. So we did a number of experiments in the rhesus monkey. And then we did with Mike, the first in utero transplant using a maternal stem cell as a donor. But again, also in this situation, although the patient was a SCID X1 patient, had a clear therapeutic benefit, it was clear to me that the engraftment was insufficient to restore the immune system. And it was clear that there were bottlenecks that we were facing that we didn't anticipate it when we thought that the fetus would be tolerant to every transplant and that the fetal stem cells would be even more tolerogenic compared to the adult stem cells. So what was the issue? The issue was, first of all, space. If we don't create space for the hematopoietic stem cells of the donor or the autologous engineered stem cells, you don't have sufficient engraftment. Second issue was the allogenic immune response. So the fetus was perfectly equipped to reject unless it was a severe combined immunodeficiency. And the rejection was mediated very early stage by innate immune responses, but also by adaptive immune responses that are established in human fetuses as early as 14 weeks gestation. And as soon as the thymus is populated, we have an immune response where the immune system knows very well how to recognize what is self and what is not self. So from these wrong assumptions that we had, that the fetus was tolerant and that the fetal stem cells were permissive, I and my team decided to focus on three major possible ways to overcome these barriers. One is the antibody-based non-genotoxic conditioning. And I'm not going to talk about this because I know that Agnieszka will cover this field in a beautiful way. The other one was a way to induce tolerance to the allogenic hematopoietic stem cells to overcome the barrier of graft versus host disease mediated by the donor T cells or rejection mediated by the host T cells. And the third one was actually from a discussion with Paul Berg that he said, what about gene therapy, Marie-Grace? Why don't you overcome your problem by using the autologous hematopoietic stem cells instead of the allogenic and engineering the autologous cells? So this is really history now. But to give you an idea how we move from, hypothesis to reality, first of all, I can tell you that 20 years later, we know exactly how tolerance is established. There are two major mechanisms of tolerance. One is through the clonal deletion in the thymus, which occurs when we give purified stem cells that migrate to the host thymus. And the second is peripheral tolerance mediated by regulatory T cells. And indeed, today we know that there are different types of regulatory T cells. The best characterized in the mouse and in the human are the FOXP3 Tregs. We distinguish the thymic-derived FOXP3 Treg that emerge from the thymus with a defined set of TCR antigen specificity, the mostly recognized self-antigens, or the peripherally induced Tregs, mostly in the gut, that recognize non-pathogenic antigen and are induced in the peripheral tissue. But by studying a tolerant patient transplanted with fetal stem cells, we discover also a new population of regulatory T cells that we call regulatory type 1 cells. At that time, I was working at DNEX, and Bob Kaufman and Tim Mossman were discovering the Th1 and Th2, so I thought to be creative to call them TR1. Now, we never went to a TR2, unfortunately. But TR1 cells are unique because they don't need FOXP3 for their function. And IL-10, interleukin 10, which was cloned actually from the cells of these patients, the human IL-10, is the master regulator for these TR1 cells. And what we know today, which we didn't know at that time, that these regulatory T cells play a key role in maintaining immunological homeostasis and preventing pathology. So autoimmune diseases are prevented through regulatory T cells, but also allergy, chronic inflammatory diseases, and fetal maternal tolerance is mediated by regulatory T cells. So these regulatory T cells can indeed be used as a drug, as a living drug, to induce tolerance in patients that receive an allogenic stem cell or organ transplant. And the whole concept is that if we give these regulatory T cells, we will induce tolerance and eliminate the need for chronic immunosuppression. And my team at Stanford is working on both cell therapy with FOXP3 Treg and TR1 cells. Rosa Bacchetta indeed is developing the drugs with FOXP3 Tregs, but we have also in the clinic the TR1. And of course, because we are pediatric allogenic stem cell transplanter, we decided that the first product, the first TR1 cell product developed in the lab would go to prevent graft-versus-host disease in children receiving an allogenic stem cell transplant because of hematological malignancies or genetic diseases. So we have now ongoing a trial, actually two trials, with TR1 cells to prevent graft-versus-host disease. I just want to remind you that graft-versus-host disease, which is mediated by the donor T cells, is still a major cause of morbidity and mortality. And we lose still many children, many patients, because we cannot control this disease, which is a systemic disease in the gut, in the lung, in the epidermis. And even when we give massive immunosuppression, once the patient develops severe GVHD, it's really the chance to have a good outcome are very slim. So we have now a trial in which we give a product that we call TR10 cells, highly enriched in TR1 cells, to prevent graft-versus-host disease in patients receiving mismatch related or unrelated hematopoietic stem cell transplant. We indeed have two trials ongoing. In one trial, we give the cells a day -1. In another trial, we give the cells after stem cell engraftment. We have treated so far 10 patients. I think that this is a very important proof of concept to show that the cells we give are safe and can modulate the allogenic response. Of course, this is very preliminary. We have now a follow-up of 1 year only in 3 patients. What I can tell you is that in these 3 patients, we can trace the cells we infuse up to 1 year. And I don't need to go in the detail. So this is very preliminary, but it's good news, because if this cell therapy works in children receiving allogeneic stem cell transplantation, you can imagine that we can use the same cell therapy to induce tolerance in the fetus. So these trials will show that we have a safe product that can be infused in patients without any side effect and with efficacy in inducing tolerance. But what about gene therapy? Well, when we started to work in gene therapy to cure genetic diseases, the field was really very, I would say, immature. So we were using retroviral, gamma retroviral vectors to transduce the stem cells in vitro. And what we were transducing was a combination of stem cells and progenitor. And this gamma retroviral vector had a very low efficiency of transduction. We transduced around 10–30% of the stem cells. And also, in the very first trial conducted in Paris, they showed that these gamma retroviral vector were not safe because they induce insertional mutagenesis. You know, what we do with this viral vector is to a gene addition. So we integrate the healthy gene in the genome of the cells in a random way. So with the gamma retroviral vector, what we found very early on is that the integration was preferentially close to gene and also close to oncogenes, which led to what we call today insertional mutagenesis and leukemia transformation. So we quickly moved from the lentiviral vector, from the gamma retroviral vector to the lentiviral vector. But nevertheless, with the gamma retroviral vector, we developed and got approval for marketing authorization of the first stem cell gene therapy product for severe combined immunodeficiency due to adenosine deaminase. It's a genetic defect due to the deficiency in an enzyme, which is very important in the differentiation of the T cells. So the lentiviral vector, which is what we use today as really the first option for autologous stem cell gene therapy, are safer than the gamma retroviral vectors, and they have a higher gene transfer efficiency. So back in my previous institution in Milan, we developed several cures with this lentiviral vector. One already reached approval and marketing authorization, was licensed to Orchard, and Orchard got approval under the name of Libmelday, is stem cell gene therapy for metachromatic leukodystrophy, and the other one is stem cell gene therapy for another primary immunodeficiency called Wiskott-Aldrich syndrome. So far, so good. What we are developing at Stanford is stem cell and T cell therapy for a rare genetic disease, which is not so rare as we thought at the beginning, which is due to mutation in the FOXP3 gene. We recently got approval from the FDA, and we are starting a clinical trial in which we correct the T cells of the patients with lentiviral vector encoding for the FOXP3. So this trial will show us if we are able to correct the regulatory cells leading to prevention of systemic autoimmunity, including inflammatory bowel disease and type 1 diabetes. And this is the work of Rosa Bacchetta and her team. But the other thing that we learn over the years is that this lentiviral vector gene therapy can be really beneficial for a number of diseases, but is also a major also has major limitations. One limitation is, of course, the lack of physiological regulation of the transgene expression. The other limitation is the potential risk of insertional mutagenesis, also with lentiviral vector, which, although they are safer, the gamma retroviral vector, they can still potentially lead to transformation. So we really think that the future for stem cell gene therapy is gene editing. And of course, we are very fortunate to have a Stanford Matt Porteous, who is a pioneer in gene editing, and he developed a system in which we really can correct the typographical error by removing the defective gene with CRISPR-Cas9 and adding the healthy gene, the healthy DNA by AAV transduction. So we have now a trial that was moved to a spin-off company, to Graphite Bio, in which we do the gene editing for sickle cell disease. So with a single substitution to correct the typographical error. So it's a single mutation in a single gene. And of course, this first trial will open the door to many other trials using this technology to correct genetic diseases of the blood and the immune system. We are also working very, very actively in using this gene editing technology for IPEX, for the FOXP3 mutation, as well as for other genetic autoimmune diseases in which we think that fixing a single gene can lead to restoration of immunotolerance. So we are really in an exciting crossroad in gene therapy, because not only, sorry, I think I did something wrong. I don't want to start from the beginning, don't worry. We are really at the crossroad of bringing this new technology to from the bench to the bedside. I just want to remind you that the Center for Definitive and Curative Therapy was established for all of us that want to translate our discovery from the bench to the bedside. We have now a beautiful GMP facility where we can do the process development, the scale up, and the manufacturing of the cells. We have also a dedicated team that can support the investigator with the IND filing and running the phase 1 trial. And as you know, we were very fortunate to have a gift from the David and Lucille Packard Foundation to build the 5th floor of the new hospital, where we have dedicated beds for phase 1 trials. So we have investigators from pediatrics, from derm, from medicine, from many departments that can treat their patients in these beds. We are allowed to treat children and adults up to 45 years of age. So I think this is really a unique infrastructure for all of us to support our translational research and our phase 1 first in human proof of concept trials. And with that, I would like to thank my lab and of course, all my clinical colleagues in the stem cell transplantation regenerative medicine section, Rajni Agarwal, who is the PI in the Tier 1 cell therapy trial, and the Center for Definitive and Curative Medicine, Matt Porteous and Tony Oro, the co-director of the center. And I want to thank you for your attention. Thank you.
Thank you so much, Maria. I know a lot of people probably have a lot of questions for you. I found it fascinating that you came back full circle to now be in the Fetal Symposium, where you started your career, and hopefully you don't come out of this saying, oh, fetal therapy is a failure and we shouldn't pursue this, but rather the other way around. But
time is right. The timing is right.
Timing is everything. Timing is everything. So thank you. We're equally excited about having the next speaker with us, which is on a similar but as Maria alluded to, a little bit of a different angle looking at these types of disorders. So Dr. Nishka Chekowitz is a clinical hematology oncology expert, but she's been a physician scientist and a clinician scientist for decades, and an entrepreneur, and has spent a lot of time in trying to understand how to improve hematopoietic stem cell therapies, predominantly in children, but we're very excited that she's starting to think about fetal therapy, and is actually a very sought after expert in symposiums related to fetal hematopoietic stem cell therapy. So we're really excited to have Agnieszka here. We're really excited about her presentation and hopefully generating some more questions and collaboration. So Agnieszka, I'll let you take it from here.
Thank you all for the invitation to present today. It's a true pleasure to be joining this group, and it's just super exciting to be seeing all of this being built here at Stanford, and quite a feat of work to put all of these things together. So congratulations on what you guys have built so far, and we're looking forward to contributing to this in the future. So let me just try to pull up my slides here. And I'll try to build upon some of the wonderful work that Maria Grazia has done and talked about during her presentation. So I'm going to be focusing on our efforts on our progress towards safe curative treatment of blood and immune diseases and enabling treatment at really any developmental stage, including fetal therapies. Really, we're building upon the foundation of many individuals and decades of work here, but focusing on hematopoietic cell transplantation, which Maria Grazia so beautifully introduced during her talk, which is this very unique procedure that really allows us to cure many different blood and immune diseases. And this isn't just a theoretical procedure, but it's something that we do in our stem cell transplant unit every day, not just at our institution, but also around the world. And this procedure relies on being able to use healthy hematopoietic cells, often from a donor, but as Maria Grazia also alluded to, we could also use gene-corrected cells, and using these hematopoietic cells to then transplant them into a diseased patient, which allows them to then recreate a new blood and immune system that can persist for the life of the patient. And we've done this in many different settings as a field, and many different diseases and different age groups, and over 1 million patients have now been treated with these types of hematopoietic cell transplantation procedures today. And these procedures can really be used to cure many different blood and immune diseases because they rely on the power of the hematopoietic stem cell, which is this very unique cell that has the ability to self-renew for life and also give rise to the full blood and immune system. So when you start thinking about how many different blood and immune diseases are out there, the list really can get very long, as there's a lot of different ways that this complex system can fall apart, both due to genetic defects or acquired defects. And on this slide is just a list of some of the many different diseases that can be caused by defects in hematopoietic stem cells, or diseases that can be cured by hematopoietic cell transplantation. And this list can even be further expanded if you think about manipulating hematopoietic stem cells to have additional function on top of their native function as well. But while we can do these types of procedures today, and many patients have benefited, we do have quite a few limitations to these types of hematopoietic cell transplants. On the left is just a graphic schematic showing the different steps involved in this process. And this is still at a very high level. There's many more details that are involved in this. And what you can see is that it's complicated. There's a lot of parts that have to go into this. And then on the right are just 3 of the main complications that patients can experience: graft versus host disease, serious infections, and gross tissue toxicity. And sometimes these complications are actually worse than the diseases that we're treating. And so it's both the combination of the complexities of these procedures, as well as the potential toxicities that limit us from using stem cell transplants today in all of the different disease indications that they could be used in. And especially it's these combination of issues that really limit us in the use of these types of transplants in the in utero setting. And as Maria Grazia alluded to, one additional piece is this conditioning piece, which is even more challenging to do in the in utero setting. Today, in order to get a hematopoietic cell transplant to engraft in a patient, we have to give patients very high doses of chemotherapy and radiation, which to an adult patient causes issues, yet alone what it might do to a fetus. And so conditioning with traditional approaches is not really feasible in the in utero setting, but we've been working on trying to develop improved conditioning approaches that can be used at all developmental stages, primarily focusing our initial efforts on adults and children, but hoping to get more into the in utero setting, which I'll talk about as well. And it's really this conditioning that I believe causes a large majority of these complications. It's the conditioning that triggers a lot of inflammation, which then triggers graft versus host disease. The conditioning disrupts the mucosal membranes and specifically allows infections through the skin and through the gut. And it specifically also causes the gross tissue damage, such as infertility, hair loss, organ damage, such as cardiac and liver toxicity as examples. And so our goal for these types of hematopoietic cell therapies is to dramatically change the way that we do them, to really have 100% survival and 100% cure rates and stop talking about survival curves in our field, and to really also eliminate all of the toxicities associated with these procedures, both the short-term toxicities and the long-term toxicities. And my research efforts have been really focused on the replacement of the toxic conditioning, which, as I mentioned, I believe strongly contributes to all of these complexities. And I was very fortunate to join Irv Weissman's lab as a graduate student here, where we started to explore the use of conditioning and developed this concept of using HSC targeted conditioning as a replacement to this broad-based genotoxic conditioning that is currently used. In a series of experiments that we did when I was a trainee, we actually showed that your own hematopoietic stem cells compete with donor hematopoietic stem cells. And rather than ablating the entire marrow or ablating the progenitor cells, all one needed to do to get effective engraftment in at least the immunocompromised setting was to get rid of one's own host hematopoietic stem cells. And so we started developing these approaches that can do this in much more targeted means, specifically using antibodies, which can be extremely specific. And by doing this, can decrease the toxicities and the collateral damage to other tissues and organs. In initial work that I did as a medical student here, we showed that using an antagonistic antibody to this important receptor on mouse hematopoietic stem cells known as CD117, we could deplete the host hematopoietic stem cells in a mouse after just a one-time treatment of this antibody. And then we were able to show that, excitingly, this type of antibody-based conditioning approach could now allow for donor engraftment of hematopoietic stem cells without the need for chemotherapy and radiation. And we no longer saw just very small amounts of engraftment, but we're able to get extremely high amounts of engraftment with just this type of antibody-based conditioning. And then as a postdoctoral fellow in Boston, I continued to build upon this approach and make additional agents that were even more powerful. And specifically, we worked on developing antibody drug conjugates, which could be used at smaller doses with increased potency, or around the same idea of depleting host hematopoietic stem cells even more powerfully, enabling even higher amounts of donor engraftment post-transplantation of donor cells. And so we've been very excited about advancing these types of efforts, given that we've been able to show that single agents can so specifically deplete host hematopoietic stem cells and then enable very high donor engraftment post-transplantation. Excitingly, we've also shown that these types of approaches are very safe. So not just theoretically are they safer, but in actuality, when we've looked at all of the tissues of these animals, we haven't seen any toxicity apart from some mild cytopenias with the antagonistic antibody in extremely high doses and some mild grading of the fur, again, in extremely high doses. And then with the antibody drug conjugate, just some mild elevation in the liver transaminases, but nothing compared to what we see with traditional chemotherapy or radiation based conditioning. And based upon these exciting preclinical study results, there have now been several efforts that have spun out to develop clinical equivalents of these approaches that are now either in clinic or advancing closely to clinic. And so we've been working with these various partners to continue these translational efforts. And so we see a number of different opportunities to use these types of antibody-based conditioning methods. One is to combine them with gene-corrected autologous cells and use them as single agents in that setting. The other is to combine them with immunosuppression to prevent immune rejection and then be able to transplant allogeneic cells in immunocompetent patients. And so also being able to use this approach in settings where autologous gene-corrected cells are challenging to make. And then we see opportunities to use these types of antibody-based conditioning approaches really at all ages and at all stages, including at fetal therapies. And through some collaborative efforts that we've done over the years with colleagues that have expertise in the fetal transplant space, we've actually shown that these agents, or at least the preclinical versions, can be very effective in these settings. This is just some data that we did in collaboration with Tippy McKenzie's group at UCSF, where we showed the initial anti-CD117 antagonistic antibody that we developed could be used in utero and actually deplete host hematopoietic stem cells in the in utero setting in pregnant animals. And then we showed that when we transplanted these animals at birth, we could get significantly enhanced engraftment of the donor cells and correction of various different diseases. Also in collaboration with Bill Parentow's research group at CHOP, we were able to show that if one did an unconditioned in utero transplant, one could establish some baseline level of chimerism. And then when we did an antibody-based conditioned transplant afterwards as a single agent, we were able to significantly boost that. So that was an alternative approach to be able to use these antibody-based conditioning approaches to establish very high engraftment after an in utero manipulation. And so now we're really in the waiting stages of waiting for these antibodies to be further developed and to really get FDA approved before we can use them in the fetal setting. So currently there are clinical trials being run in either adults or in certain rare genetic diseases. And our program here in particular has been involved in clinical trials with the first generation antibody in severe combined immune deficiency in adult patients with MDS and AML. And we're in the process of opening up a clinical trial in Fanconi anemia that I'll mention briefly as well. And then we're also working in the interim on further improving in utero allogeneic transplantation in the mouse setting and looking at alternative approaches for safe and effective autologous gene correction. And in these latter two programs, we're in particular focusing on this disease, Fanconi anemia, which is a disease that's very close to my heart, as it's a complex disease that can have pretty grievous side effects in these patients. It's technically a multisystemic disease, although it primarily affects the hematopoietic system, and it's caused by gene defects in one of 23 different genes that are involved in the Fanconi complex. And unfortunately, these patients, by having mutations in these genes, have ineffective Fanconi complexes, which makes them hypersensitive to DNA damage. And these patients can have certain congenital abnormalities, but the biggest problems that they tend to face is that almost all of these patients develop bone marrow failure and have a very high rate of development of cancers. And so over 80% of these patients develop bone marrow failure by the time they're 12 and require allogeneic transplant. And although our treatments have gotten better and better to be able to take care of patients once they develop bone marrow failure or leukemias, we have still a lot of room to go. In particular, these patients still have pretty significant short-term complications, develop pretty significant mucositis, have to stay in the hospital for weeks to months. And then unfortunately, they also have significant long-term complications. And the most severe of which is that almost all of these patients end up developing secondary malignancies, which makes sense because we have patients who are hypersensitive to DNA damage, and currently we give them chemotherapy and radiation, which then accelerates that process. And what you can see in this graph here on the right is that really almost every single patient who has received a transplant ends up getting a solid tumor. And right now we don't have any great ways of treating those patients. So they're kind of ticking time bombs, and we induce even bigger problems in these patients while having to correct their hematopoietic issues that develop earlier in life. And so optimally, we'd be working on several different ways of improving this treatment process for these patients. One would be diagnosing these patients as early as possible, so we can be monitoring them more carefully and then potentially intervening even earlier, optimally in the in utero period. We'd also be diagnosing them earlier, as I mentioned, to decrease complications and decrease familial anxiety, as often these patients today get diagnosed only when they have the development of bone marrow failure or cancers, and that we'd also be treating them without any short-term or long-term toxicities. And right now, most of our efforts have been focused against the hematopoietic problems that these patients have, because that's the major issue. But in an optimal world, we'd also be working on correcting some of their other cells and other tissues, which with the advent of gene therapy may be a possibility in the future. In collaboration with the Stanford Center for Definitive and Curative Medicine that Maria Grazia built and alluded to during her beautiful presentation, we have been focusing on this disease already in the pediatric and adult setting. And in particular, we've been working on running 2 novel clinical trials over the last several years for these patients, which has given us a wonderful opportunity to attract these patients to Stanford to better learn about their disease and then to offer them these novel treatments. One is a unique gene therapy trial that we've been running in collaboration with Rocket Pharma that I'll tell you a little bit more about in subsequent slides. And then the other is for patients who are already in bone marrow failure, where we've been trying to replace that genotoxic conditioning, and in particular, the total body radiation that we currently use, or the busulfan chemotherapy with the first generation antibody that I mentioned that came out of our efforts here at Stanford. In terms of the gene therapy work that we've been doing, this is a high-level schema of the treatment protocol. What we do for these patients is try to identify them as early as we can before they develop bone marrow failure. And then we give them mobilization agents that consist of G-CSF and plerixafor. And we cause their stem cells in the bone marrow to really get pushed into the peripheral blood. And then we apherese them from there and collect them from the peripheral blood. And then working with our commercial partner, Rocket Pharma, these cells then get enriched and modified with a lentiviral gene therapy vector with the FANC-A gene. And so this specific treatment is only available for patients with the FANC-A subtype, which is about 60 to 70% of these patients. And then these patients get treated with their own gene-corrected cells. Because of the unique toxicities of these patients to traditional genotoxic chemotherapy, we've been treating these patients without any conditioning, which is pretty unique to this disease setting. And excitingly, what we've seen in the first few patients that have been treated has some pretty promising results. So there are 3 patients now that at the last interim follow-up had over a year of study data. And these are the 2 patients with the most promising results, where we show increasing vector copy number, stabilization of blood counts in these patients, and resistance of their bone marrow cells to DNA damaging agents. Unfortunately, one patient that was in this first group of patients did develop a bone marrow failure after she got an influenza B infection and has done well after an allogeneic transplant. But these other 2 patients have not needed to undergo allogeneic transplantation and exposure to those genotoxic medications, which has gotten us rather exciting. And given these results, we've now expanded the study and treated more patients, which I won't be mentioning about today in the context of this talk, but it is really exciting that this treatment is on the path to approval for these patients. But in the context of this talk, what I really wanted to focus on here is just to show that the 2 patients and that we have the longer term data that have not yet developed bone marrow failure and hopefully never will, have had increasing vector copy number in their peripheral blood over time. And this is pretty exciting for 2 reasons. One is that hopefully these cells continue to increase and persist for the life of these patients and really cure them and prevent them from having any hematopoietic problems. But the other is that it really shows us in these patients that these gene-corrected cells likely have a survival advantage to the normal, to the Fanconi anemia cells that are not corrected. And that these now more normal cells really have the ability to outcompete the Fanconi anemia cells, which has given us some ideas for additional types of treatments that we can do in these patients. And this phenomenon has also been observed in preclinical models by others and in this natural process or in this spontaneous process known as natural gene therapy, where there have been a couple of handfuls of patients that have developed somatic mosaicism and actually had stabilization of their blood counts and evolution and expansion of the corrected cells in a similar manner. And so we're hoping to now take advantage of really this competition or this knowledge of competition of wild type cells over Fanconi anemia cells and use this in the development of novel fetal therapies. And in particular, we've been collaborating with the Stanford Department of Obstetrics and Gynecology and through support of the Dunlevie Maternal Fetal Medicine Center for Discovery, Innovation, and Clinical Impact to start this preclinical research effort to try to explore fetal therapies for this disease, which hopefully then could be expanded to other potential diseases as well. In particular, we're interested in improving fetal diagnosis of Fanconi anemia, which, as I mentioned, could be useful in then potentially treating patients at this stage. But if not, then at least putting them in a position to be able to have the gene therapy in the pediatric setting prior to development of bone marrow failure. And then specifically, we're working on 2 projects to explore fetal therapies in these patients. While we're waiting for the antibody-based conditioning to continue to evolve, we're also exploring doing in utero hematopoietic cell transplants in Fanconi anemia models without antibody-based conditioning, given this phenomenon of wild-type cell competition over the Fanconi anemia cells, thinking that this might be the ideal setting to treat hematopoietic diseases without conditioning. And then we're also starting to explore in utero gene correction with a number of different collaborators to see if we can use gene therapy to either improve the hematopoietic system or other tissues as well, which I think might be mentioned during the next talk. So with that, I'll just pause and thank the many different individuals that have been involved in these efforts. There's been many individuals here at Stanford and throughout the world that have been very, very helpful. And this is just a picture of our research lab and some of the specific individuals that have been working on these many different efforts. It's a heroic feat to launch these types of clinical trials, and we're very grateful to the CDCM and our clinical trials program, where Grazia, my colleague Rajni Agarwal, and many others that are involved in these efforts. Thank you very much.
Thank you. Thank you so much, Agnieszka. I did see one question in the chat, but we'll save it until the end, Virginia, until the next speaker. And that'll give us some time for some questioning and also another break after the Q&A. So bear with us for another 20 minutes or so. We're really thrilled to have Kyle Cromer as the final presenter of this session here. And Kyle's a PhD who works in the Proteus lab and has been spending a lot of time thinking about not just in utero genome editing, but genome editing in general, and especially its in utero applications and vectors and target disorders. And he's really on the cutting edge of this entire field. And while Agnieszka spoke a little bit about this, I'm sure Kyle will be able to expand and get us all thinking about gene editing in the future and what we can do today as well. So Kyle, thank you so much for joining us. We really appreciate it. And we look forward to your presentation.
Okay, thanks so much. Thanks to the organizers for having me. I'm going to be giving both parts of this presentation. It was originally going to be a joint presentation with Carson Charlesworth for the second half. And so he gets all the credit for the data generated from that. So what I'll be talking about is really the workflow that we went from an allo-HSCT treated disease to something that we can potentially treat using genome editing as perhaps a one-time curative in utero intervention. So just a bit of background before I go into this is alpha thalassemia is actually surprisingly common. 5% of the world's population harbors an alpha thalassemia variant, and this is largely due to the selective advantage it imparts against malaria, similar to how sickle cell disease this variant does. And the rates of disease are thought to be rising due to population growth, human migration, and advances treating milder forms of the disease. It's been largely overlooked for gene therapy because it's historically been lethal in utero, but because of these recent advances, intrauterine blood transfusions allow survival to birth. And then at birth, if survival occurs till birth, then they're treated like beta thal or sickle cell disease patients and treated with frequent transfusions and or iron chelation therapy. The only potentially curative option is bone marrow transplant from a matched donor. And this is, as Maria Grazia mentioned in her talk, it's limited by availability, risk of immune rejection or graft versus host disease. As Agnieszka just mentioned, significant morbidity due to myeloablation protocols, current regimens, and then it's only possible postnatally. This therefore indicates a major unmet medical need. And there are analogous therapies that have been developed for beta thal and sickle cell disease using lentiviral vectors or genome editing. And we believe that we can use similar approaches to develop a modern curative treatment strategy for alpha thal. And so this is building from work that we just published for beta thal. What we did was use the Proteus Labs platform of CRISPR-AAV6 mediated genome editing in HSPCs ex vivo. and we can edit a patient's own HSCs and transplant them in, these corrected ones, to overcome the shortage of limited availability of donors or immune rejection or graft versus host disease. And so just very briefly summarizing this work in one figure, we fully replaced a copy of alpha globin with a transgene, with the beta globin transgene, to restore hemoglobin tetramers in these patients. Alpha thal, however, has in the most severe instances, alpha thal major has lost all copies of alpha globin, leading to accumulation of beta globin dimers. This causes two problems. This loss of alpha globin is too much beta globin causes these toxic dimers to form in the absence of their binding partner, alpha globin, that transport but don't release oxygen. And then also the absence of alpha globin causes severe anemia. So using an approach modeled after our beta thal correction strategy, what we'd like to do is use CRISPR-Cas9 to introduce a cut in one of the alleles of beta globin, and then use an AAV6 DNA repair template that could fully replace the HBB endogenous locus with an alpha globin transgene, therefore restoring hemoglobin tetramers and improving the beta to alpha globin imbalance in the context of alpha thal. In essence, one editing event would address both problems. Too little alpha globin is solved by alpha globin knock-in, and too much beta globin is solved by a knockout of one of the copies of beta globin. And so I'm going to talk about this in terms of design, build, test cycles. The tools right now are good enough that we can start to apply engineering principles in living cells. And so this is just a workflow to go from 0 to 100, essentially. How do you start by designing a genome editing strategy? And so the very first step is to understand your constraints. We want to screen CRISPR guides to understand the regions that we can induce breaks, that then, therefore, we could introduce high frequency corrections. What we wanted to do is, operating within the constraints of this disease, we wanted to introduce break sites outside of the exons of HBB so that an indel alone, in the absence of homology-directed repair, would not disrupt beta globin production. And so we screened guides in introns 1 and 2 and the 3' UTR in collaboration with the Bruce Conklin's lab at UCSF, and we found 3 guides that cut at high frequencies. And so we essentially have ruled out some of our one of our possible strategies of targeting 3' UTR. We can proceed with these other 3 guides to the next design build test cycle. This next cycle is screening homology arms based on the constraints from the previous cycle. So guide RNA 7 cuts in intron 1, and then our 2 other efficient guides cut in intron 2. So I designed AAV repair vectors that constitutively express GFP so that we can rapidly assay editing frequencies mediated by cut sites in intron 1 or cut sites in intron 2 with corresponding homology arms. The right homology arm immediately flanks the cut site in both instances, and then the left homology arm immediately flanks the sequence upstream of the start codon so that we can knock in a transgene in the clinical vector into the start codon of this beta globin allele. And so what we see is that we were able to achieve moderate editing frequencies, and that actually targeting intron 1 is the most efficient, likely due to the shorter distance at which we're splitting off these homology arms. And just a note, the 200 base pair intervening sequence in this intron 1 donor would be lost and replaced from homology arm to homology arm with our integration cassette. So the next step in this design build test cycles are to screen UTRs in promoterless vectors so that we can understand the kinetics of the expression when we knock into the start codon here. Because these are promoterless cassettes, they're promoterless with alpha globin, HbA1 or 2, and then a 2A YFP reporter cassette so that we can understand the kinetics by which they turn on over the course of erythroid differentiation. We wouldn't, because beta globin is only turned on as HSCs differentiate down this lineage, we would expect not to see green cells if we've achieved red blood cell specific expression until we get towards the end of this protocol. And so what we see when we track the kinetics is that all of these cells edited with all 3 vectors and a constitutive GFP vector all differentiate fine, but we don't see, as we expected, GFP turning on until later in the process, as is the case for all 3 of these vectors that we designed. So this is really reassuring. And in this heterogeneous population that we've differentiated, we do still have some residual progenitor cells. And so what we also want to see is that in the, well, that was the bulk population, the GFP percentage that I just showed. But what I'm showing here is that in cells that retain progenitor markers, we see no YFP expression. However, ones that have lost 34 and 45 and gained CD71 and GPA, these in vitro erythroid markers, they're highly expressive of GFP. And so this is assuring us that we have actually achieved red blood cell specific expression. And this is something of a red blood cell specific safe harbor site. And so what we see here is that we have the most efficient editing achieved with the vector with HBA1 UTRs. And then we also have a vector with HBB UTRs as well as HBA2. And then the expression level of our HBB UTR vector is highest relative to HBA1 and HBA2. And so this essentially gives us 2 winners to proceed to our 4th design build test cycle. Again, this is built from the intron 1 integrating scheme with that intron 1 targeting guide. And so then to test clinical vectors in patient cells, which we're right about to do, the next step was to take the previous vectors, the winners from the previous round, again, this intron 1 integration scheme, remove our GFP cassette, our YFP cassette, so that they could be clinically relevant. And then we want to target wild type cells first to ensure that we're able to target at high frequencies, which indeed we are. And so the very next step that we're going to be doing in the next month or so is to edit patient HSCs, which we've received from UCSF with a collaboration with Tippy McKenzie. We'll quantify editing rates, ensure that red blood cell differentiation is unperturbed. And then in these cells that have been differentiated into red blood cells, we'll perform single globin and hemoglobin tetramer HPLC to ensure that we've been able to restore in vitro hemoglobin tetramers in the context of alpha thal. And then we want to perform mouse transplants to ensure that edited cells are capable of long-term engraftment. This is, as I've already alluded to, a highly collaborative effort with Don Cohn's group at UCLA and Tippy McKenzie at UCSF. They're essentially doing the analogous approach for a modern curative alpha thal correction scheme. Don is integrating lentiviral alpha globin expressing vectors, analogous to Bluebird's strategy. And then Tippy is performing a CRISPR screen to look for guide RNAs, essentially cut sites that de-repress zeta globin. And so this would be analogous to CRISPR therapeutic strategy for beta thal. And then as such, as a part of this highly collaborative effort between the 3 labs, we're in a unique position to compare all 3 methods. CRISPR AAV6 platform in the Porteous lab, which I just showed, the CRISPR MDEL approach that's being developed at UCSF, and Don Cohn's lentiviral delivery, and what Don Cohn referred to as a bake-off. And I think that this will be really a powerful comparison for the field. Our immediate goal would be to file an IND for ex vivo alpha thal correction with the best approach. And then the future goal would be to adapt the best correction strategy for in utero intervention. So this now brings me to the data generated in collaboration with Carson that he would have presented if you were available. He gladly allowed me to present this. And so what we've done to start getting towards editing in vivo that we could then apply some of these ex vivo correction strategies towards is to establish a mouse model. Taking cue from the Amy Wagers group at Harvard, we're using the AI-14 mouse model, which has a constitutively expressed CAG promoter leading into a stop cassette flanked by LOXP sites, then leading into a TD tomato. So in the instance of CRE delivery to these cells, the stop codon would be removed, and then we would see TD tomato expression. The first step of this is to optimize in vitro, just some of the basic parameters here. So we did this to mouse HSCs, the AI-14 mouse HSCs in vitro. And just these simple comparisons, what promoter works best in terms of expressing CRE that we then could use for later approaches. And so we see that SFV is better than CMV. We see that a BGH poly A tail is more effective than an SV40. And then we also see very dramatically that a self-complementary AAV is much more effective because it allows us to bypass the rate-limiting step of second strand synthesis involved in AAV transduction. It's much more effective than single-stranded. However, packaging capacity is much more limited. And so the optimal vector, though there are some constraints with this, would be self-complementary, SFV expressing with termination initiated by BGH poly A. So that's great for our in vitro optimization. So taking some of these lessons that we've learned from that, now we wanted to look in vivo at targeting. So we did retroorbital injection of AAV6 and then analysis 3 weeks post-injection. And what we found is that single-stranded, and the liver is just a great first pass because we're able to target this at such high frequencies. So single-stranded SFFV expressing CRE with BGH is about 50%. We got about to 100% with our self-complementary vector. However, we also found that the source of our AAV mattered a lot, probably because different purification methods remove empty capsids. So we found that one source for Vigene, even though it was suboptimal in being single-stranded, CMV, and SV40 terminated, it also elicited 100% transduction with this CRE delivering AAV6 virus in the liver. What we can also do with this mouse model is to model in vivo genome editing. And so what you can do is take an SA-Cas9, which allows us to be, is small enough to be packaged into an AAV6 vector with LOX-P targeting guides. And so then when both guides cut at the same time and the stop codon is removed, we can get TD tomato expression. However, there's a dramatic drop off in the efficiency. So we know delivery, so CRE is great at assaying delivery, transduction in vivo, but then we have an issue with activity upon transduction. So what we are able to see some in vivo transduction using an SFFV SA-Cas9 vector. So this is a nice proof of concept, but certainly requires much more optimization in this pass. And it also may be improved by this Vigene prep, which this was not, which we saw a dramatic increase with the CRE vectors. But we don't really care about liver in this case. At least postnatally, we want to make sure we're able to transduce HSCs. And so we did retroorbital injection of these mice and actually found that using immunophenotypic HSC markers, the SLAM panel, we're able to transduce about 6% of HSCs in vivo. We're actually able to edit at least this TD tomato expression from LOX-P excision for SA-Cas9 at 1%, which might be enough to cure certain diseases such as Fanconi anemia because there's such a selective advantage lent to cells. Then just one quick example of some of the optimization we can further do with this mouse, and then take back to the lessons we've also learned previously and combine this optimized protocol. We tested different injection sites, retroorbital versus intrafemoral versus tail vein injection in the caudal artery. We found that caudal artery delivery is significantly more efficient at transducing HSCs than retroorbital, and then also intrafemoral seems to give us a boost as well. So we can use these optimizations in future experiments. The next steps would be to further optimize the system in a variety of ways. One of these would be to combine this with the mobilization regimen, delivery of our editing reagents, which has been shown to boost both viral and non-viral mediated HSC transduction. So we're really optimistic this could also make our HSCs more accessible to transduction and editing. We want to titrate the dosage by balancing HSC transduction with toxicity and immunogenicity. We also, as we've seen, the purification method / commercial vendor matters a lot. And then towards taking this that we've learned by targeting in vivo postnatally, we think that in utero targeting would have some real advantages. So such as the open fetal circulation, accessible stem cell populations in the fetal liver, naive immune system, and the small fetal size are all very distinct advantages of especially in the context of a disease like alpha thal that already requires in utero intervention. And so in that case, then we would want to adapt all of these ex vivo approaches, perhaps to correct diseases in HSCs in utero, and begin with diseases when corrected cells have a selective advantage, or alpha thal where they require in utero intervention. So that concludes my talk. Thank you, everyone. And I have to thank Matt, Tippy McKenzie, Don Cohn, and Karsten primarily, but a lot of other people on the slide too. Okay, thank you.
Thank you. Thank you so much, Kyle. It's always tough just to give your own presentation, let alone somebody else's presentation. But you did a great job. It was very smooth. I know we're running a little bit behind, but I promise you will be done by 8:30 as promised. And I think just given the quality of these presentations and speakers, I don't want to detract from this Q&A. So I'd like to spend or give people enough opportunities because you know, it's very rare for us to have such amazing presenters and at our, you know, fingertips and be able to target them now with questions. So if anybody has any questions for our speakers, please, now's the opportunity, and no need to raise your hand. You could just unmute yourself. If not, maybe I'll just start unless somebody else has one. My question is to Maria, Grazia, and Agnieszka. In terms of fetal therapy, what do you see as the ideal donor for these hematopoietic stem cells? Is it some sort of modification of the mother? Is it a different donor? Is it what's your thought there?
Agnieszka, why don't you take it?
I was going to say, I think you have a lot more experience.
Okay, then I go. Well, okay. First of all, of course, as we heard from Kyle, the perfect donor is the autologous, if we can. In principle, I would say that if we do a gene therapy approach and we cannot generate sufficient autologous cells, because that would be a challenge in the fetus, maybe, we can think about an HLA match, sibling, or an HLA match unrelated. If we think about an allogenic donor, not genetically engineered, but really to replace the fetal hematopoietic system, again, the ideal donor is an HLA identical. But we have now, thanks also to the technology established by Alicia Bertaina, the opportunity to use haplo, mother of father, where we deplete from the T cells and the B cells, what we call the alpha beta TCR B cell depleted transplant. And this transplant in the allogenic setting has the advantage that there is very little GVHD because we deplete of the donor T cells with the alpha beta TCR. We have the gamma delta, which are not so powerful in mediating graft-versus-host disease. So I would say if you have an HLA identical donor, that's ideally either allo or engineered. If you don't, I would go for an haplo purified. I don't know if Agnieszka agreed with that.
I do agree with that. But what that really opens up is the opportunity for every patient to have a donor. And so with these types of strategies, we're really looking at a variety of different approaches that could work, but something should be available for everyone, especially with the haploidentical donor options that we've now opened up with these newer technologies. And in the fetal setting, that means the mom or the dad potentially.
But the question for Kyle, correlated to what you say, is that do you think that if you go for a gene editing approach for alpha thalassemia, you will be able to recover sufficient number of autologous stem cells, or you have to go for an HLA identical sibling?
I mean, that's a great question. I don't think we'll have much insight until we start to, I think, get closer to taking it to patients. I mean, one of the competing technologies too that I'm also rooting for is the transplantation of maternal HSCs as an in utero intervention that Tippy McKenzie's is leading in a clinical trial. And so that could leapfrog the necessity to do this elaborate genome editing. But I think it's still, you know, there's certain diseases where that might have distinct advantages over genome editing. Or if we could deliver the genome editing reagents direct to the fetus, there's no concern about how many cells do we pull out and transplant back in.
So I personally think that that's the way to go. If you go in utero, you have to go in vivo, because otherwise you will never recover sufficient number of autologous stem cells to give back. So the in vivo approach is what needs to be developed.
There's also some really interesting work in our field of people taking iPS cells and turning them into hematopoietic stem cells and making iPS lines that are HLA matched of all types, and potentially having banks of HSCs that one could then use that could be very effective in this type of setting. Further away, but within the realm of possibility here.
Yeah, I have a question for the panel. This is Virginia Nguyen. I'd like to get your insights into how you think the immature immune system of the fetus can be taken advantage of for the in utero therapy. Or do we really think about the fetus the same as we think about pediatrics and adult patients?
Yeah, Virginia, that was the wrong assumption I had. I thought that the fetus would become completely tolerant to allogenic stem cells, and indeed is not. The only situation in which we see acceptance of allogenic cells is when the fetus is immunodeficient. And actually, in that situation, when you have maternal engraftment in the SCID fetus, these babies are born with graft versus host disease. But an immunocompetent fetus, like in the case of thalassemia, has NK cells as early as 12 weeks gestation. And we have to calculate that in the human fetus, the thymus gets populated around 8-9 weeks. And by 12-14 weeks, you have T cells in the thymus, which are TCR rearranged. So the innate immunity is there from the very beginning, and the adaptive immunity, of course, these are naive cells, but the adaptive immunity is there as early as 14-16 weeks. So the assumption that I had, which was totally wrong, was that the fetus was tolerant. The fetus is not tolerant at all. The mom is tolerant because these microchimeras in the mom of the fetal cells induce a sort of tolerance in the mom, but the fetus is not tolerant at all.
The one thing that I'm excited about with more targeted approaches to conditioning is to tease apart what we're trying to accomplish. And so a lot of the conditioning that we do today kind of does two things. It gets rid of the stem cells, it gets rid of the immune system, it gets rid of a lot of different types of immune cells when you're using cyclophosphamide, fludarabine, very non-targeted approaches. But as we're really looking at what we need to get rid of and developing more targeted approaches, I think there might be ways of really almost doing surgical dissection of the cells that you don't want using drug therapies. And so potentially taking out those NK cells very specifically using safe targeted agents together with the stem cells using a different set of agents to kind of come up with what's the right cocktail that does exactly what we wanted to do rather than these sledgehammer approaches. And if that cocktail is much safer, then you could do it much earlier in the developmental period.
All right. Well, thank you so much. And I think maybe we'll offer people an opportunity to ask questions, after the symposium as well, that maybe we can pose to the speakers, individually. We'll work on that because I know time is short and we have so many outstanding presentations. It's 7:37. We do have two absolutely superb speakers for the last session, and we will be able to get out of here on time, but I want to make sure we get a break. So it's 7:37. Why don't we regroup at 7:45? And that should give us enough time for the two speakers and the last Q&A and final thoughts. So please come back, and I promise it'll be fascinating presentations, and we'll see you in a couple of minutes.